First of all, I would like to thank Alberto for accepting the invitation to this online seminars, the BGP series. Hopefully, in the future, we can have again the presential seminars and meet together and have some beer and the lunch that is obligatory to have. So Alberto studied environmental science in the University of León, and after that he did a PhD in biotechnology and biomedical applications in the University of Valladolid, studying the splicing regulatory sequence of two genes, BRCA1 and BRCA2, by hybrid mini genes involved in cancer. Among his major achievements and contributions, I would like to highlight two of them that I think is very important, more for us, is the co-foundation of two agrotech companies. One is AC Gene Reading Life, and the other one is Biomarkers, that is involved in the analysis of microbiota for plants. He has also received more than 20 honors and awards, mainly related with technology and innovation, such as the top 15 Scaling Ambition Agrotech companies in 2020. And today he's going to talk about the use of microbiomes and the properties as markers to infer the health of the soils and the potential use to improve plant growth and productivity, I guess. So thank you very much, as I said, Alberto, for accepting the invitation. After the talk, Aaron will open the microphones and you can ask the questions to Alberto. Thank you.
Thank you, Juan Carlos. Thank you very much for all the people that is attending to this webinar, to this call. Today, I'm happy to introduce new concepts in the microbiome world, especially related with how to approach microbiome studies, not only from the taxonomy point of view, if not for the ecological point of view, and how we can use that to improve agriculture, sustainability, and soil health in agriculture. That is one of the goals on the missions of Biomakers as a company, as a startup. Let me share my screen. Yeah, this one. Please confirm that you can see the slides. Because right now okay, perfect.
Yes, we can see it.
Okay, well, yeah, as I mentioned before, the mission of Biomakers is using microbiome as a biomarker for a more sustainable agriculture, especially linked with soil health and also with new way of practices in agriculture, from regenerative agriculture concept, organic agriculture, but why not, for example, traditional agriculture, but adjust specifically the management practices? Why not? And also consider that life is essential to grow crops, especially in soils, and because the human population is growing and people need more and more food, it's very important to take care about soil. But take care about soil is take care about biodiversity in soil. That's our idea on how to approach that. And also, if we are thinking about sustainability in agriculture, it's very common to link the concept of soil health to the sustainability. If we go to the USDA, and because our company, Biomakers, is a US company based in Sacramento, we are following that specific concept from the USDA that define like soil health, like the capacity of the soil to provide a specific functions to promote living ecosystems and sustainable life. This means that only living things can have health, and the living things in the soil are microbes specialized in relation with the plants to provide a specific insights that allow the plants to grow. It's in the same way that humans, in our gut, there are millions of microbes that allow us to receive the nutrients from the food that we are eating. In agriculture, it is more or less the same. Plants are colonized for microbes in the roots, for leaves and other parts of the plants too, but essentially in the roots that allow plants to interact with the environment, adapt to the stress, for sure provide nutritional functions, but are linked also with specific disease problems or other factors. Well, if we are taking a view about the soil microbiome, we can identify millions of microbes per gram of soil that are living in like a specific ecosystem. This is a specific picture that we get from electronic microscope in 2015. You can see here the surface on a plant, can be the roots, can be the fruit, different things, and 8 white plots or dots are specifically microbes, no? The surface of the microbes. And I like to see that, okay, there are different microbes that in an ecosystem, like a community, like building specific communities. And I like to compare this specific picture that we can observe in the microscope with the, for example, the surface of the earth and how humans are habiting the earth in different communities, cities, and other things. If we want to study without any reference, because soil microbiome is like a new science right now, thanks to the DNA sequencing and other technologies, how to try to understand that communities, the relation of that communities and how that communities are interacting with the plants. That is a key question, and there are different approaches to try to understand first and then apply these microbiome indicators. First of all, we can take a soil sample and we can run a culture in a culture media in the lab. But the problem or the issue to do it is that maybe less than 1% of the species that are living in the soil can be grown on a plate. It can be the first issue. It's not possible like to see directly with our eyes how microbes are interacting. We need to study in a different way. The last 10 years, because the development of DNA sequencing specialized, allow us to introduce these kind of technologies in order to isolate all the DNA that we can find in a soil sample and sequencing that DNA and try to assign that piece of the DNA to a specific microbes. In order to understand what are the taxons, the species that are living in that soils, in order to understand the functions, the genes that that species have in the soil, and especially try to understand how that gene complexity is working together to provide functions to the ecosystem, and also to understand the relationship between one microbes to others. If we talk about different approaches that are using DNA to explore microbiome, there are 2 specific techniques that we can use: Amplicon sequencing and shotgun sequencing. The basic difference between Amplicon sequencing and shotgun, with shotgun, we are isolate all the DNA and we are sequencing all the DNA without differentiate nothing. All the DNA that we can detect in the sample. Depending on the number of reads that we get per sample, we can get more or less information from that soil DNA, from that microbiome community. For the other hand, it's very common, because of cost, apply Amplicon sequencing. And it's, for example, the kind of service that regularly we are offering in the company. There are 2 different divisions in Amplicon sequencing. First, from the methodological point of view, applying short sequencing or long sequencing, because there are different sequencing platforms. Each platform, each technique have different issues and benefits. Depending on what we can do, we can select one or each other. And related with amplicon sequencing, there are other divisions. We can amplify specific genes linked with taxon, so to explore the taxonomy of the sample. For example, 16X is well known that it can be applied to the bacteria identification of taxons, bacteria kingdom. Or for example, ITS is the other marker, for sure there are others, that can be used for the identification of fungal communities in the soil. But the other division is that we can use amplicon sequencing to target specific genes that are involved with specific functions. For example, functions linked with the nutrition of the plants, nitrification, or others, okay? For that, there are different options. There are, one can be benefit for one thing, another for others. Related with the taxonomy resolution, using short amplicon sequencing, we can get most of the genera that is present in the sample and also the species. But we can play with OTU levels, operational taxonomic units, or SBA, that is the new name for a similar concept with more resolution of clustering. For the other hand, using long reads, okay, we can go from the species level, also strain level, and using SATGOM strain level for the more abundant species that are present in the soil is possible, because we can get for that species, more or less the whole genome of that. This is related with taxa. Taxa is very interesting to explore biodiversity. It's very interesting for understand the known species versus unknown species that for sure we can identify in the soils, and we can classify in the tree of life with less or more accuracy. For the other hand, it's linked with the functions. Using amplicon sequencing, we can predict the functions, and prediction is more or less in a correlation between 70% and 95% with the data that we can get for SATGOM sequencing. There is a lot of advance in this field. There are at least 3 different softwares available for researchers that develop these kind of tools, and it can be useful. There are people that believe a lot in the predictions. There are people that don't believe on that. But a part of that, the data say that the correlation can be good, specialized in soil. But these kind of techniques were developed specifically for human gut microbiome. For the other hand, with long reads, yeah, we can be more precise, and also we can use, as I mentioned, specific gene targets linked with functions, linked with a specific process of the microbes, and is not predictive, is real data or real quantification of genes that are present in the sample, but we can only target a few of them, not all of them. With SATGOM sequencing, we can target all of them. We can identify the genes, but we don't know what is the expression of that genes if we are not using metatranscriptomics. For the other hand, ecological point of view. From the ecological point of view, amplicon sequencing can allow us higher resolution than SATGOM, because we are targeting specifically genes that allow us to identify not only the more abundant species, if not all or more or less all the species that can be involved in the samples. In a resolution similar to the qPCR, between 100 or 1,000 cells per milliliter, is more or less the resolution that we can get with amplicon sequencing in a deep sequencing, okay? More than 300,000 reads per sample. For the other case, for SATGOM, we need a lot of reads. We need more than 30 million of reads to get a good resolution in a sample that have a high biodiversity in the soil, and also the influence of the plant genomes, animal genomes into the counting, for example, prokaryotic species, is a big issue, because the genome of that species is very, very large for that, compared with the prokaryotic, and that can influence a lot in our analysis. Okay, this is the basics of the techniques that we can use for targeting to study microbiome. Today we are talking about Amplicon sequencing short reads only applied for taxonomy. Okay. Well, if we want to explore microbiome, the first thing is understand if microbiome can be a good or bad biomarker and for what. Especially the first sentence that we can say is soil is a reservoir of plant microbiome. This is the preliminary study, the study that I follow in the beginning when we start to work in microbiome field. This specific study comes from a researcher named Irache, from Basque Country, but when she worked in Chicago with Jack Gilbert. They explored the influence of bulk soil microbiome in the plants, especially in vineyards. And the conclusion of that study is that there is a connection between bulk soil and soil lithosphere that are more similar between them. And also there are links between the bulk soil microbiome and other parts of the plant. In this, today, you can see a lot of principal component analysis where each plot is a sample, and the distance between the samples represent how similar or different are the microbiome and taxonomic and abundance of that taxons in the sample. It means that all samples that here is grouped have more similar microbiome than the sample that are far in other color. I don't know if you can see well the picture, but in general, the picture represents that the soil microbiome influence in the root microbiome because they are more similar, and then microbes are colonizing the different parts of the plant. That means that soil microbiome is the best or the reservoir of that communities in the plant. Then we were exploring that in our database because we started in study vineyards. We have a lot of data from that specific crop. Here is a representation of our soil microbiome database in comparison with grapes and wine. Plots are representing taxons, and if the taxons are present in soil, but then in grapes and wine, and we can see that samples that we are identified in the beginning of the fermentations, before applying any specific treatment to the wine to avoid the microbes, 79% of the microbes that we identify in the soil were present in the wine. That means that the influence of the soil microbiome in fermentations and also in grapes are high. Okay. And also, okay, that's for vineyard, but what happened for other kind of crop? This is another question that is always present in our mind. The things that we can learn in one crop can be applied in other crop. Well, this is a study that we performed in Mexico 3 years ago, where we study bulk soil microbiome, root microbiome, and also other parts of the plant, including leaves. And here is the PCOI that is representing the samples. You can see here in yellow the plots linked with the roots, with the soils. Sorry, with the soil in pink, with the roots in yellow, and other parts of the plant. And also we can see here again that the soil microbiome is very, very similar to the root microbiome, and is connected specifically with the plant microbiome. And why we think that the soil is the origin of the reservoir? It is because the more abundant species that we can find are always present in the soil. Then similar abundance can be present in the roots, and then the abundance decrease. This is an alpha diversity analysis for fungal species and for bacteria. Well, now we know that the origin of the plants that can, in relation with the plant, is the soil microbiome. But soil microbiome can be affected for different factors. This is another interesting thing to explore. Is all the microbes that are present in the soil are always the same microbes, or there are differences between the different factors that can affect to the microbes? It's something that the people is exploring, research including us, how microbes are affecting for climate, soil characteristics, different physicochemical components, and also the humans, the management. No? And the conclusion here is that soil microbiome is a biochemical good biomarker. But that specific factors influence in different way in the microbes that are living in the soils. This is a paper from Noah Feurer in Nature that explains at the global scale the factors that are influencing the microbiome. Soil pH, organic carbon, percentage of redox are the key specific factors that are affecting to the microbiome. I can confirm that our data more or less say the same. There are other factors, for example, that are more linked with the climate, like moisture or, for example, temperature of the soil, that can influence a lot in the microbes that are living in that soils. But again, soil texture or the level of different nutrients in the soil are affecting to the population that we can observe in terms of taxonomy and also in terms of abundance of that taxons. But life that is affecting to the microbiome, specifically the kind of crops that we are growing in the field, is not the same the microbiome that can be linked with grapes that the microbiome can be linked with the corn. Each plant are producing specific chemicals in the root that attract specific taxons of the interest in the plant, like us in the gut microbiome. Also, other, for example, viruses like us, no, with the coronavirus, can have a big influence in the microbes that are living in the soils. Well, here today we can, when we started to explore that idea or that specific concept, we launched a specific analysis that right now is available in preprint in collaboration with Denmark University. We're taking more than 400 different samples from different vineyards in the world at the different locations, different latitudes, different pH, different soil conditions, different weathers. And we try to explore that analysis. And one of our conclusions was that the regionality is very important. No? All that factors, soil texture, climate management, can be reflected in a regional level, for example, at country level. And you can see here, like an 16S and an 80S, a bacterial and fungal analysis, the samples are clustering per location at country level. This is like the terroir concept at macro scale level. But if we go in deep, for example, at regional level, for example, in Spain, we're taking samples from different regions that typically are growing vines and produce wine. And also we can have the same conclusion. Samples are grouped per a specific region very clear. Okay, regionality is a key factor for that. But this is a global level. What happens if we take in samples from 2 different countries? We launch a random forest analysis, for example, with more than 500 samples from US and Spain, and try to predict if we receive a sample and we don't know the origin of the sample, what is the accuracy in the prediction at country level from what is the origin of that sample? Well, with the random forest analysis, we can predict with 94% of accuracy what is the origin of the sample that we are receiving in our labs. That can be great because that factor, soils, structure, climate, location in general, can be very linked with the taxons that we are identifying in the samples. But today we are talking not about the regional or the factors that can influence in the communities, if not the management. Well, looking at this, if we are putting different samples with different management in this kind of analysis at taxon level, it's not possible to understand if a sample is under a conventional management or an organic or biodynamic management. For that reason, we take, for example, we take only soil samples from US. We run a principal component analysis, and we then try to differentiate organic and conventional management. And it works. Working at regional level, especially when more regional level you are working, better resolution you can get in order to understand if microbial communities are under conventional management or organic management. But it's not possible then to compare with samples from other regions because the taxons are depending on the location. If this is clear, we need to explore other metrics in the microbiome to understand the influence of the management into the microbial community. Sorry. Okay, other relevance part of the why taxons are not the perfect biomarker to identify different management practices is that this is the biomakers database in last year. You can see here a principal component analysis of different samples, thousands of samples, coming from different crops and different locations. And what we can observe that samples are grouped not only by the region, if not by crop. Crop influence a lot in the taxons that we can get, locations too. That's two factors. But how to study management practices influence in soil microbiome, to use microbiome as biomarker on that. Our approach was avoid taxons, and let's start to think about communities. Imagine if we can explore how are the communities or how that communities are working in different locations, we can explore the social relationship between the species of taxons that are living in different locations. And that is independent of the name of the taxon. Let me explain, because we explore a network analysis into the microbiome in the same way that social media can be applied for other proposals using network analysis. Imagine that we want to explore how is a city without knowing what people is present in the city. Well, I can analyze my network. I, for example, like to go to the gym. I'm going to the work. I'm going, for example, to practice some sport. And in that specific place, I have in relation with other persons. That, in general, in the gym, there are persons that have a link with me. No? We like to practice swimming. For example, if we go to the work, I'm in relation with the people that are always in the office. And this is other different people that our common thing is that we are working in the same way, in the same company. Well, why not to apply that concept to the microbes, independently of the taxons that are present, to understand how are the relationship between one microbes and others. For example, if we want to understand why the number of police in Tokyo is less than the number of police in Spain, in Madrid, I need to put the police and the cities in a context with the different persons with different functions that are present in the environment. Okay, we apply these kind of concepts to the microbiome in order to explore if these kind of concepts can avoid the location metrics, no, that are linked with the taxons in the microbiome, and explore the influence of the more close influence, like human influence, in the soils in microbiome. For that, imagine if I can define my specific network, my social network, then I can define the social network of others. Imagine that we taking a sample in a city, no, and in that sample we have a picture of the social, of my social network. But if we take different samples from the same city that are representing the networks of the different persons, then we can build a meta community that allow us to understand how that city is working and the kind of clusters, the kind of functions that are performance in that city. In the beginning, maybe I don't know the functions or the clusters are related with functions, but I can see the cluster, I can measure the cluster, I can measure different metrics. Well, that's the approach. From specific local networks. It means the networks, the co-occurrence of the species in a negative and a positive way that we can detect per sample, we can start to build meta-communities in order to understand the probability to detect one specific taxon. If one specific taxon is present in a sample, what is the probability that other taxons are present or not? And we can create positive, co-occurrent communities, and in the other way, we can calculate co-exclusion. If this microbe is present in the soil, what is these other microbes never are present, and this is because there is a negative ecological relationship between that microbes for any reason. Okay, but that's the origin of that concept. Well, we have local networks and we build a meta community. From the meta community, we can then compare each local network with in front to the meta community, and we can start to get some specific data. For example, modularity. Samples, when they are comparing in front to the meta community, can easily select different like groups of samples, group of taxons in the same sample, or sometimes we cannot observe that modularity. We can start to understand the density of the network. There are networks that are involved a lot of different taxons. There are networks that decrease on taxons. And also it can be related with the beta diversity, but in a different way. There are these kind of properties that specific models, specific nodes, specific connections are metrics that we can measure from the specific microbial communities. And why it can be related with the management? Well, we launched a specific analysis with, I think, 400 different samples. Half of the samples come from Spain, from different locations, vineyards. Other samples come from US, different locations too, especially Napa, because it's the more representative samples that we have in our database, with different management practices, classifying conventional management practices, or organic management practices, and biodynamic management practices. And we build meta-communities, one meta-community for Spain, another meta-community for US, and also a global meta-community, combine both Spain and US. Here is the clustering. At taxonomic level, Spanish soil microbiome taxonomy are different to the US specifically taxonomy. For that reason, we divide the meta-communities in two, and then we combine. Here are the representation of the meta-communities, okay? But consider that taxons are totally different. Well, we, after get the meta-communities, we start to explore the metrics linked only with the management practices, because here it's impossible to see what are the specific conventional samples or organic samples. But we explore modularity, clustering, and co-exclusion proportion in the networks. And look at this. It's very easy to see that with low level of modularity is linked with specifically biodynamic samples, also for both Spain and US. Unconventional in Spain is more similar to organic, but in US, conventional practices are very well differentiated from the organic practices. Only looking at the microbiome network properties. At the same time, we can compare the clustering, the level of clusters. Looks like if the clustering, here we can see differences too between conventional and organic practices. And again, in Spain, curiously, conventional and organic are very similar, but very far compared with biodynamic samples. And here is using two communities, and here is combining the two communities in only one. And the results and the conclusions are the same. What that means that now we can use microbiome indexes, microbiome properties based on networks that allow us to differentiate very well and different management practices. Well, and this is how we can visualize their properties and we can differentiate the samples. This is a biodynamic sample with a lot of different species that are interconnected with each other without creating clusters. And the density of this community is very high. That we can interpret that this community is more resilient than others. Because if something happens, if we introduce a chemical, a fungicide, a bactericide in the soil, okay, some of the microbes can die, but all microbes are interconnected, and the resilience, or hypothetically, we can understand that they can survive to that specific change. For the other hand, in the conventional management, we can see clusters, and that clusters of microbes that are performing or are grouped because they have similar functions, like I mentioned before, no? When I go to the gym, I'm in relation with some specific persons with the same interest. It is the same. These microbes are related with each other because some specific interest. And in this case, why they can be very different? Because the human pressure, here we are adding chemicals, NPKs, fungicides, products into the field in a more hard way than in biodynamic, that is like more natural agriculture, more linked with the forest, no? We can say. And especially here, human is pressured to the microbiome to perform some specific functions, no? And here the problem is that if something happens and break a group of microbes, the other microbes can be not linked with each other, and the probability to survive for that community can be compromised. In the middle, we can see organic management that is like in the middle of the properties of them. I represent US samples because in Spain, organic looks very similar to conventional. That means the conventional agriculture can be more similar to organic because of the different regulatories that in agriculture that are in US and Spain. Well, this is not something that only we can observe. Here I present another study that is comparing, for example, conventional agriculture with non-tilling practices and also organic. And the conclusion were specifically the same. The number of nodes, the number of different interactions that we can see in the field, and here is a different crop, this is soybean, is lower in the conventional agriculture compared with non-tilling and organic. And modularity is higher in conventional related with non-tilling and organic. It means that others with similar approach have the same results. And the conclusion can be that specifically network properties can be used to define the impact of the management practices into the soils, independently of the taxons, independently of the location, independently of the kind of crop. For the other hand, it was very interesting, other study for Marcel that take some specific soil samples, get the microbial community and pass that samples from a different physical surface that can decrease the number of microbes that can then put in the plate, I think, or in a specific lab experiment. And if they take all the community in order to decrease physically the number of microbes that can be present, then we measure the network properties. And it's very curious to see that the network properties that we can measure under these specific conditions change over the different pressure that we can get, in this case physically, on the microbial communities. And looks like the sterilized community are more clustering, okay, less density of microbes. And I like to compare conventional agriculture with this kind of analysis, because look like biodynamic organic can provide more functions or more diversity of functions, have less specific modularity. And for the other hand, in conventional, right now, we can have an equilibrium of functions, but that equilibrium is very thin and can decrease because any factors that can influence in the microbial community that we can perform. For that reason, well, based on this data, we can rethink the agriculture and also try to define what we want in order to preserve that complexity in the soil, that biodiversity that can provide specific functions and resilience, sustainability too, in the ecosystem. I also link that with soil health. Okay, again, microbiome as a biomarker of management practices. Microbiome networks can be linked or to explore disease risk. Because we play with 400 samples from different locations in vineyards, we then try to understand if there is a link on the network properties and the presence and abundance of pathogens linked with the vines in that specific vineyards. And we find a very nice correlation. More biodynamic is, and the network properties linked with that biodynamic properties or organic properties, more abundance of pathogens we find. But for the opposite way, under conventional agriculture, less abundance of pathogens we find. But it is not linked with the risk of the disease. It's in a positive way. Think about a hospital. In a hospital, the abundance of microbes or bacteria that we can find is very low. The probability to find key disease bacteria for human is very low. But if there is someone present, the risk of disease for humans is very high. In the same way, we can think about that specific results. Conventional agriculture, where we can apply hard fungicides, okay, the probability to find that specific pathogens in the field is very low. But if we find someone's, the risk to perform a disease can be in very high proportion. Then, of course, we can explore other concepts. Okay, we can analyze the management practices in a big level, but why if we analyze specific product applications into the field? In the company, we are offering a service that we can analyze the effect of any kind of ag input into the field, into the soil microbiome. This is an example of a product that we test, that is a biological product, that we see that in some specific locations, the product is affecting to the microbiome after the application of the product when we are comparing with the similar locations without applying the product. In this case, it's a the principal component analysis show us that there is a big impact in only one location of the product, also was in relation that that specific impact was linked with an improving of 40% of the yield in that location. And here we can see the metrics that we calculate in order to understand that the effect of the treatment compared with no treatment was higher in this case compared with the others. Also, we measure the network properties and we find that in this location, the network properties were affected. Why not try to understand if that change in the microbiome can be linked with the yield, if we can model that? We explore taxons and we find specific taxons because it is very clear in principal component analysis that is linked with the taxonomy and abundance of species. We find taxons that specifically can be explained or linked with that increase of the yield, but also we explore network properties. And we run a random forest analysis in order to understand if these metrics can explain the yield. And also, that's not the future. We were working on that direction in the last 2 years. 2 years ago, we built the first predictive model based on network properties. Last year, we launched specific trials in order to improve that prediction with more locations, more data. Right now, in this year, probably during the March and April, we are recruiting people to explore or receive samples from one specific crop, one specific product, in order to predict or test our predictions in the product recommendation and also in yield improvement prediction. This is thanks because we have a very ambitious project right now that started last year. It's named Fields Forever, that we are in collaboration with 73 different universities and companies around the world, working on the different specific projects, testing biostimulant, carbon sequestration, yield, disease, and that project is ongoing. I aim to all people that is attending to this call that they have some project or is interested to participate in some of these projects, that contact with us, because we are happy to increase the number of partners for that proposals. Well, thank you very much. Again, this is not like only science. The good part of the startup is we need to run fast, especially to survive, but also because we are approaching the market very quick, and we are applying science in order to translate microbiome properties, not only networks, taxons, and functions in specific reports for diagnostic what happened in the soil and what is the status in the soil, but also to apply to test what is the effect of the different products and why products, specifically biologicals, prey and probiotics, have a very nice effect under some specific conditions or locations, and in others it's not possible to see that effect, and how microbiome can help us to explain us and build models in order to recommend the best products for the best fields, increase yield, get sustainability, and improve globally soil health. Thank you very much.
Thank you very much, Alberto. It has been a very, very nice talk, and has been very stimulating to see how we can translate science into economy and society. So now we can open the turn for questions. Aaron, can you reactivate the microphones? So if you want to ask a question, just activate your microphone and ask the question to Alberto, please.
I cannot believe that there is no question.
There is no questions?
I have a question.
Okay, go ahead.
Hello?
Please
Hello?
say your name, because I can't see the name.
Okay, hello, I am Soledad Sacristan, and thank you very much for your talk. And I think I did not understand very well. So when you are building these networks, you are not taking into account the co-occurrence of the OTUs, but you are taking into account the co-functionality of the OTUs, or how is it? done?
First, we can play with the co-occurrence of the OTUs or SVAs. Okay, that's the first. One of the issues is that if, as most samples that you put in the meta-community, more difficult is to build the meta-community. And also meta-community can be clusterized, okay? But yes, we are including the co-occurrence, inclusion and co-exclusion metrics to build that meta-community. And then after that, we are comparing 8 samples in front of the meta-community to get the properties of 8 samples in relation with the meta-community.
So you hypothesize the functions of the community based on the co-occurrences of the OTUs.
No, first is to understand the properties, no, and start if there are clusters of not, only based on taxons. After, see if the community is producing clusters like in the conventional agriculture, then we can explore what are the properties, functional properties that we are inferring to all the samples or all taxons that are grouped under some specific cluster, in order to understand why that taxons are combining together or are working together in some specific function, and try to understand that functions. It's like, imagine that you are taking or building networks from different persons in a city. If you don't know how is organized the city, you can see a cluster, but you don't know if the cluster is linked with the cooking classroom or the gym classroom. You don't know. Then we can try to ask to the people that are building that cluster, what are the common things that they have to perform some functions.
Okay, thank you.
So, Alberto, I'm Antonio Molina. I will put my camera just to. So I was surprised, or maybe not, about the small difference between the network of between organic and conventional vineyards in Spain. So do you guess that this is related with that some of the, let's say, most used pesticides or fertilizer are considered also as organic pesticide fertilizer, like for example copper, because this may explain because copper, I mean, is highly used in vineyard, as you know, and is considered in some legislation and also in the Spanish one just to be organic compound.
Yeah, that's.
I mean, organic something that you can use in organic farming.
Yeah, that's one of the hypotheses. The first one is that the different regulation in different countries can allow us to understand why conventional agriculture is more similar to organic, especially in Spain. Maybe in France it can be similar. Also, we can explore maybe that when we are talking about vineyards are in different places. Vineyards are not very big in general, no, like some hectares, and they are very close to each other. Sometimes we can see that people is taking a block like, okay, this is organic or biodynamic, but the block close to the other is in a conventional agriculture. It was an hypothesis that we tested in France. We ran an analysis from a specific client in order to try to differentiate biodynamic place very close to other blocks that are under conventional and transition. But the result was that was very easy to identify the biodynamic place compared with each others that are in transition or conventional. But it depends. For the other hand, we are in touch with farmers all the time. When you are approaching about certification, or we are talking about certification of vineyards, it's about papers. Okay, certification of the papers is the kind of product that you are adding to the field. If something happened 1 year, okay, high level of pressure of mildew or idiot or like I said, we know perfectly. My family have vineyards, or other people that I know, friends have vineyards, and sometimes they decide to add specific products, including if they are under organic management. Here we explore that we have a metric that we can use, a part of measure the chemicals that are present in the soils, that can be related with the management practice that you are using. For the other hand, we have a lot of clients that are thinking into, are under conventional agriculture that want to go into the organic. And sometimes for certification, you need 3 years, 5 years to get it. Why not to get that in 1 year? It's possible or not, depending on the level of the status of your block in the beginning. And now we explore the idea to measure that level of the interaction of the ecosystem in a field, and explore the evolution of the new practices that you are adding into the field in order to understand if that it can be more fast or quick getting that specific organic fingerprint. But yes. Also,
okay.
we don't know what's the effect of other products that people is considering, like, for example, organic products, no? For example, probiotics. If you add a specific bacteria with a fungal activity, okay, you are reducing probably or pushing the specific microbes in one direction because you are reducing biodiversity, but maybe it's less aggressive than others. We don't know. It's something that we like to explore, and probably here in the call there are a lot of researchers that have that specific question.
Okay, thanks.
Okay, are there more questions?
Hi.
Hi.
I'm Beatriz Garcia. I'm interested in the evolution of the microbiome over time that Alberto has explained at the end of the talk. I would like to know, have you checked if the network changed over time when you change the management practice? I mean, could you move a soil from a high to a low modularity, for example?
Yes, that's the good part, that we are testing a lot of different products over the time using control place and treated place. And we can see that that is possible. But depends on the location that the evolution is less or higher, okay? Sometimes it's nothing. Again, it's the same with the, similar to the taxons. Taxons are evolving because of the time or the season, and can be linked with the plant phenology, or can be linked with the weather conditions or physical conditions like temperature or moisture in the soil. But if we look at the taxons, in general, taxons change in abundance, but we identify more or less the same taxons in the same location. With the network properties, is different. It is because we can see if that taxons can be organized in a different way. Okay, and in general, that needs a change in the taxons too, or change in biodiversity. We decrease or increase the number of taxons that we are observing. But yes, the answer is possible. Also, we have nice observations with no-tilling practices and cover crops in France in corn, that is possible to see over the time that they are that practices are improving the network properties. In the case that we can say improving, okay, changing.
Okay, thank you.
Okay, I think we have another question from Sandra. Sandra?
Yes, hello,
Okay.
Can you hear me? Okay, hello, my name is Sandra. And well, indeed, I have a lot of questions, but I will limit to one. So my question is if growers are asking your company to use these models or these microbiome studies to to understand the risk of a disease, or if they have a disease in the field, to understand how, or to understand the disease, and also to use your predictive models in order to avoid this kind of disease with different products. I mean, for example, a bioinoculant or this kind of products.
Yeah, in general, our clients, and right now we have 700 different clients from 34 different countries, from small farmers to big farmers. There are like always 4 or 5 different questions. First, risk of disease, but they don't want to know if they have a disease, because when they have a disease, they have a problem. They need to anticipate and evaluate the risk to have a disease. For that reason, the attitude of the farmer is, okay, I need to add a product because I don't want to have a disease. If I have a disease, I have the problem on the field, and that can be reduce the yield or other things. No, that is a problem. They need to anticipate problems. That's the key of RAM predictions, and they are using us to explore that. Why? Because most, not all, okay, but most of pathogens, specific fungal species, the reservoir is the soil. Other, maybe bacteria, other can be insert, can be mobilized for the earth, but in general, we can understand the risk that the farmer can have because the present abundance of the ecology of the sample that is sending to us. And it's working very well in some specific crops, in some specific diseases. In others, for example, so in more of the case, root or plant disease. If we go to the fruit disease, it can be a bit different because other factors like weather can conditionate a lot the prediction. But yes, that's the key of the farmer. Farmer in general predict the disease, reduce inputs because to be more cost effective in the production, increase yield, that's other metric, improve sustainability. That can be in more of the mind of in the mind of a lot of farmers. What kind of products they need to add? They started to ask us if the product is working well or not in their fields, or what kind of products are the best for them. And last question that right now we are receiving or feeling is in relation of carbon. It's a new topic that is on interest of a lot of clients. No, the potential to increase carbons or sequestrate carbon into the field, and how microbes can be linked to that.
Okay, thank you very much. Really interesting.
Okay, we have time for one quick question. Yep. If not, I have a question, Alberto. Based on these co-occurrence networks that you presented, do you think it's possible, like in gene networks, to identify hubs microbes or nodes microbes that sustains what could be the essential microenvironment for microbes, and you can use as a core? For?
Yes. It's a different of the concept of core, of the calculation of cores, but it's in that idea. Especially if we are under like the representation of biodynamic specific practices, no? Depending on what kind of community we want. Imagine in biodynamic practices, all microbes are present. No, it's like there is not key microbes in the environment that need to survive each others. Okay, because it's like a more realism. And for me, it's like in this kind of communities, all is in expansion. No, the genomics of that specific microbes are try to increase the number of species, try to increase of number of relations. but it's like a community that all are present. No, there is no key species, or there is a kind of no key species present, because all the species can perform all the functions. If this species disappear, other species can produce that functions.
Okay.
Under the conventional management, because the pressure, the human pressure that can drive the adaptability, of that community to the specific environment that we are pushing, in this case, yes, there are key species that allow connect group of species with each others. And there are the specific key species. Other thing that I don't mention in my talk is how that communities can be linked with yield. Because if you as a farmer, farmer know that under conventional agriculture, they can get high level of yields, more or less stable. Under organic or biodynamic agriculture, they have years with a nice yield and other years with regular yield. It's very explorative, and it's one of our questions. If we are focused on yield, what are the best specific microbiome prototype that we can get? And it maybe can be very different with the question of, if I want to be sustainable, what is the best microbiome that I need to get? Maybe we need something in the balance, no? It's a very interesting question. Today, I can say that sometimes conventional profile, microbiomes under this conventional profile, that clustering, etc., looks like are better relation with yield increasing.
Okay. Okay, thank you.
That makes sense. Okay.
That makes sense. Thank you, Alberto. Thank you very much for the talk and all the discussion that we had, but I think it's time to close the conference, and it's been a pleasure to have you here.
Thank you very much for all.
Thank you everyone for attending, and have a good day and good weekend.